Next one is classification of enzymes. So here, International Union of Biochemistry had given various criteria to classify the enzymes. So there was an old classification, but now the classification which was given by IUB was followed. So based on that one, there are some classes, and each class will have a subclass, and each subclass is divided into subclasses. So here, according to IUB, 6 major classes are present. So those are oxidoreductases, transferases, hydrolases, lyases. The 5th class is isomerases, and the 6th class is ligases or synthetases. So these are the 6 major classes which are present in the classification of the enzymes. Let us start with the first one, that is oxidoreductases. First one, oxidoreductases. So these are the enzymes which are concerned with the oxidation reaction and reduction reaction. So here it is basically related to the transfer of hydrogen molecule. So based on this one, so there are further subclasses are there. The first one is dehydrogenases. So these are the one in which the hydrogen is transferred from substrate to the NAD molecule. So let us take one example, that is malate is a substrate here, is reacting with a nicotine adenine dinucleotide, that is NAD molecule, in presence of malate dehydrogenase enzyme, and converted into oxaloacetate. And here the malate is losing one hydrogen which have been transferred to NAD molecule. So NAD have been undergone reduction to form into NADH plus H plus molecule. In this oxidoreductases, the second one is reductases. Reductases are the one which are involved in adding of hydrogen or removal of oxygen. So in this the nitrate reductase is the enzyme which is concerned with this type of reactions, where nitrite molecule is added with hydrogen to form into nitrite. So this is catalyzed by an enzyme called as a nitrate reductase enzyme. So where the nitrate molecule is converted into nitrite molecule. So here this is about the 2 subclasses which are present in the oxidoreductases. The second major class is transferases. Transferases are the one which are concerned with the transfer of the groups from one molecule to the other molecule. So here the transfer is done, transfer of various compounds is done except oxygen. And here, based on the type of compound that is transferred, here they have been categorized into various subclasses. Let us see some of them, that is transaminases. Here the amino group is transferred from one substrate molecule to the other substrate molecule. So here we can take an example as aspartic acid is one substrate molecule, is reacted with alpha keto glutaric acid, and this will be converted into oxaloacetic acid, that is OAA and glutamic acid. So this reaction is catalyzed by an enzyme called as transaminase enzyme, where the amino group is transferred from one substrate molecule to the other substrate molecule. And the second one is called as kinases. Kinases are the one which are involved in the transfer of phosphate molecules from one substrate to the other substrate, or from one compound to the other compound molecule. So like let us take some examples like fructose 6-phosphate. So this is a reaction of glycolysis, where it is a 6 compound molecule. It will react with ATP and take up the one of the phosphate which is present in ATP to form fructose 1,6-bisphosphate, and the ATP which is losing one phosphate molecule, that is converted into adenosine diphosphate, that is ADP molecules, and this is catalyzed by an enzyme called as phosphofructokinase enzyme. So here the phosphate is transferred from ATP to the fructose 6-phosphate, so it is converted into fructose 1,6-bisphosphate molecule. So these are the two subclasses which are being discussed here, which are present in the transferases. Next one is hydrolases. Third major group is the hydrolases. So hydrolases are the one in which the splitting of the substrate molecule occurs in presence of a water molecule. So here when splitting is taking place, the bonds are broken. So likewise the breaking of the bonds which is taking place in presence of water is, or that is catalyzed by enzymes called as hydrolases. So let us see some of the subclasses which are present in that one. One is peptidases. So peptidases are the one which will break the peptide bond which is present between the amino acids which are present in the protein molecule. The best examples of them are pepsin and trypsin molecules. Second subclass present in the hydrolases is phosphatases. So these are the one which are concerned with the breaking of bonds in presence of water, where let us take one example, that is fructose 1,6-bisphosphate. When react with the water molecule, that will be converted into fructose 6-phosphate plus inorganic phosphate will be released. So this is a reaction in which the bond have been broken, bond of fructose 1,6-bisphosphate have been broken, so that here the phosphate which is present on the first carbon is separated out in the form of inorganic phosphate, and this is catalyzed by enzyme called as fructose 1,6-bisphosphatase. So likewise in both of them they are splitting the bonds in presence of water molecule. The fourth major group is major class is the lyases. Lyases are the one which will split the bonds or break the bonds in absence of water or without using water molecule. This type of enzymes are called as lyases. And here let us see some of the examples or some subclasses related to this one. Those are decarboxylases. Here example where pyruvic acid will be converted into acetaldehyde plus carbon dioxide. So this is a reaction of anaerobic respiration, where the bonds which are present in the, because here the pyruvic acid is an 3-carbon compound, acetaldehyde is an 2 carbon compound. So the bonds have been broken where carbon in the form of carbon dioxide have been released. Then next one is aldolases. Here aldolases in which let us take example like fructose 1,6-bisphosphate. So it is a 6 carbon molecule. So it is acted upon by an enzyme called as aldolase, where it is converted into 2 molecules of 3 carbon molecules. So that is glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. Both of them are 3 carbon molecule. So the splitting of the bonds are taking place in absence of water molecule. The fifth major class is isomerases. So isomerases involve the conversion of one isomer form into the other isomer form, or we can call it as a intramolecular shift, in which one isomer is converted into other isomer form. Let us take some of the examples here. That is the reactions which are present in respiration and also in the photosynthesis, like glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate. Both of them are the isomer forms. So it is catalyzed by triose phosphate isomerase enzyme. One more reaction related to isomerases is the conversion of glucose 6-phosphate into fructose 6-phosphate. So here as we know, glucose and fructose are the isomer forms, which both of them are 6 carbon molecules. So this is catalyzed by phosphohexose isomerase enzyme. In the same way, 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid. So likewise here, all these are the molecules in which one isomer form is converted to other isomer form because of the intramolecular shift. The sixth major class is the ligases. So here the formation of new bonds is there. So when the new bonds have been formed, a new compound will be formed, in which new compound or the simpler molecules are converted into compound one. So that's why we call them as a synthetases. So let us take one example here. Glutamic acid, when reacted with ATP and ammonia in presence of glutamine synthase, glutamine synthetase, it is converted into glutamine and ADP plus inorganic phosphate. Likewise here, the glutamine is combined with the ammonia molecule, that is new bonds have been formed to form into glutamine molecule. So likewise these are 6 major classes which are present in the classification of the enzymes that was given by IUB. Based on the classification, that is, as we know that the classification includes classes and each class includes subclasses and each subclass includes subclasses. So likewise the classification was done in this manner. So here every enzyme is given an number, so which is represented with 4 digits. So that we call it as enzyme code. So here in that 4 digits, out of the 4 digits, the first digit represents the class to which the enzyme belongs to, and the second digit relates to the subclass to which the enzyme belongs to in that particular class, and the third digit represents the number of the subclass of that particular enzyme present in that subclass, and the fourth digit is related to the serial number of the enzyme in the particular subclass. Likewise, every enzyme will have the 4 numbers. So here let us take one example, that is glucose 6 phosphotransferase. So it is given an enzyme code as 2712. That is the first 2 represents the class to which the glucose 6 phosphotransferase belongs to, and the second digit 7 represents the subclass, and third digit 1 represents the subclass, and fourth digit 2 represents the serial number of that enzyme in that subclass. So likewise, alcohol dehydrogenase is there. So this enzyme have been given the enzyme code as 1, 1, 1 and 1. So that is a first class. In that first subclass, it belongs to first subclass, and in that subclass it is a first enzyme. So likewise, every enzyme will be given particular enzyme code. Based on that we can identify the enzyme related to its number.